thank you for the opportunity to visit with you again. I will try to dedicate the next 20 minutes or so to discussing what I think will revolutionize reproductive endocrinology and infertility care. But I will point out that the idea of using stem cells, in this case to create eggs or sperm, are being pursued in other arenas as well. For example, ophthalmologists are interested in creating amacrine neurons that could be used in the retina. Neurologists are interested in producing dopaminergic neurons that could be used to treat Parkinson's disease, for example. And of course, everybody would love to see the production of motor neurons, which could alleviate paralysis as well as other maladies. But we will discuss today the notion that one can use pluripotent stem cells to create gametes, eggs and sperm, in a way that obviously has been infeasible before. To give you an idea on the significance of this breakthrough, Science magazine last year dedicated a review to mammalian in vitro gametogenesis because they can see it coming and they deem it to be important. Similarly, the New England Journal of Medicine published a paper under the heading of Clinical Implications of Basic Research that dealt with oocytes from stem cells. So you can tell that scientific authorities across the ocean are very clear about the possibility or the high likelihood that this is coming our way, and perhaps relatively soon. I will begin with an infographic of the Nuffield Council on Bioethics in the UK, which defines the topics they will address in the coming years. Under human reproduction, the very first topic they propose to address is in vitro-derived gametes. There are, of course, many other topics that they wish to discuss and elaborate on, but as I noted, the very first subject is in vitro gametogenesis. We can probably all agree that if this comes to pass, this will constitute a disruptive technology. It is still uncertain whether it's achievable in the human, although I will show you where we stand on that. And whether or not this is a short timeline or a long timeline, of course, remains to be seen. But in the eyes of many, this is not all that far from where we are today. It is always interesting to go back to Aldous Huxley and his famous contribution from 1932, Brave New World, wherein he predicted several developments in reproduction. One of them was that eggs will be produced via ex vivo ovarian cultures. Not exactly in vitro gametogenesis, but not far. He was obviously spot on with respect to the fact that embryos will be produced, so to speak, via in vitro fertilization. He also predicted, which did not quite materialize yet, that newborns will be delivered, well, carried and delivered via ex vivo ectogenesis. As I said, he was spot on on in vitro fertilization, which is a reality we all recognize. But he really could not have foreseen the notion of in vitro gametogenesis, because at that time, the hypothesis that dominated the field was the so-called soma germline barrier hypothesis, formulated by August Weismann, professor of zoology at the University of Freiburg in Germany, who in his book, The Germplasm, A Theory of Heredity, from 1893, stipulated that the germline, of course, gives rise to the soma, and by dint of that is immortal. On the other hand, he maintained that the soma never gives rise to the germline, which was true at that time, and therefore is mortal. What has changed is our ability to start with a soma, or with a somatic cell, and guide it towards a gametogenic fate, that is to say, to produce a germ cell, and either an oocyte or a sperm. This is a process, as you probably know, which is known as somatic nuclear reprogramming. That process begins with a differentiated somatic cell, say a skin cell, which is converted by one of two techniques, which I will discuss, to a pluripotent stem cells. And that pluripotent stem cell can then be guided towards a gametogenic fate. The first technology that made this possible is known as somatic cell nuclear transfer, or SCNT for short. And it came about in 1958, courtesy of John Garden from Cambridge University, who published a seminal paper in Nature titled Sexually Mature Individuals of Xenopus laevis from the Transplantation of Single Somatic Nuclei. In essence, he began with a somatic karyoplast, that is to say, oh, something happened to my slides. Okay. That is to say, a nucleus from a somatic cell that he combined with an oocytic cytoplasm or an enucleated oocyte, which gave rise to a quasi-zygote, which developed into a blastocyst, the inner cell mass of which was used to produce embryonic stem cells, which could now be taken essentially in any direction. In 2006, a second technology was introduced by Shinya Yamanaka, MD, PhD from Kyoto University, who published a seminal paper in Cell titled Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors. Essentially, he proceeded to take a somatic cell, transfect it with select transcription factors which he identified, and he produced thereupon so-called induced pluripotent stem cells, as opposed to embryonic pluripotent stem cells. Both Professor Yamanaka and Professor Garden were awarded the 2012 Nobel Prize in Physiology and Medicine for their discovery. So in vitro gametogenesis essentially involves taking a somatic cell, for example, a skin cell, converting it either to an embryonic or an inducible pluripotent stem cells, and then guiding that stem cell towards a gametogenic fate. The beginning was a very modest one. In 2003, a paper saw press in Science, senior authored by Hans Scholler, who is now at the Max Planck Institute for Molecular Biomedicine, but at the time was a postdoctoral trainee at the University of Pennsylvania. The title of the paper was Derivation of Oocytes from Mouse Embryonic Stem Cells, a depiction of such a stem cell being shown in the right lower corner of this slide. Many papers followed suit, which time precludes us from discussion. Suffice it to say that we have reached now the point which I refer to as the final frontier, which is to say, taking in vitro gametogenesis all the way to mature eggs or sperm. Perhaps the most compelling demonstration of this possibility was provided by Katsuhiko Hayashi on the pages of Nature in 2016, wherein he reported on the reconstitution of the entire mouse female germ cell cycle. In essence, he started with embryonic stem cells or induced pluripotent stem cells. He then converted those to pluripotent germ cell-like cells, combined them with gonadal somatic cells because you always need granulosa or Sertoli cells to support the process. That gave rise to a primary oocytes, eventually to an M2 oocyte, which is also depicted on the left-hand lower side, and which upon fertilization gave rise to a blastocyst, which upon transfer gave rise to pups. In essence, Professor Hayashi took the process all the way in the rodent, from a stem cell to a living pup. Similar efforts, albeit by a different group, in this case, Dr. Saito, also from Japan, from Kyoto in this case, were reported on the pages of Cell Stem Cells last year, further reinforcing the observations of Dr. Hayashi. Clearly, what you, members of the audience, and myself are interested in is seeing whether or not this phenomenon can be imported or implemented in the human. An effort to generate human oogonia from induced pluripotent stem cells was reported on the pages of Science in 2018 by the group of Mitinori Saito. What they did is they started with human pluripotent germ cell-like cells. They combined them with somatic cells from a different species because they didn't have the appropriate human counterpart. And they allowed the gametogenic process to proceed, but were only able to get it to the oogonial stage, which is to say, they could not go all the way with meiotic initiation and all the other requirements which will lead to an M2 oocyte. So we will have to say that as we speak here today, at least, the process has been shown to be workable and essentially fully successful in the rodent, but we have some barriers to overcome when it comes to the human. When we think about the missing links, in other words, what is it that prevents us from going all the way in the human? Part of the problem is the absence of gonadal somatic cells or the simulation of their input. In other words, what are they saying to the oocyte or to the sperm as they develop? This notion of gonadosomatic cells surrounding either an oocyte or sperm is known as the niche. That is to say, the pocket of somatic cells that surrounds the germ cells and is critical for its nourishment and development. To overcome this relative shortcoming, Professor Hayashi proceeded to do the obvious, to generate ovarian follicles from mouse pluripotent stem cells. So now he didn't just generate germ cells, he actually generated whole ovarian follicles, which included the somatic component. That paper having seen press in Science last year. I would like to conclude with a brief discussion of what all of this means to us. What is the scientific and translational process? At the very least, having these, quote, artificial gametes, if you will advance science because it will provide us with an inexhaustible supply of germ cells. It will allow us to do detailed functional analysis in tissue culture and ask very specific questions. It would allow us to study germ-somatic cell interactions, which I already noted is an area that is desperately in need of new insight. But on the clinical end of things, this could contribute to the reversal of germ cell failure. Imagine that we may have the capacity to, quote unquote, cure ovarian failure, or for that matter, male dysfunction. This could potentially mean autologous gamete replacement. We could also gene edit gametes that are known to be affected by a heritable condition, potentially could be edited out and then replace a normal gamete instead of a affected gamete. This could also be helpful in the technology known as donor mitochondrial replacement therapy, wherein another woman usually contributes oocyte, but in this case, we could simply use a somatic cell to generate a gamete. It is probably too early to speculate with authority on the impact of IVG on IVF. But we can probably safely assume that if IVG becomes a reality, it will eliminate the need in ovarian stimulation and egg retrieval. And if that really is the case, we will also do away with gamete donation, perhaps with the exception of age dependence. But more importantly, IVF, which is currently an in vivo procedure, could be converted in time to an in vitro laboratory procedure. Reproductive endocrinologists will still be required to prepare the endometrium and to transfer the embryos. But much of what currently constitutes the busy work of IVF may well be bypassed. Whether or not this will improve affordability of the procedure, which tends to be expensive, definitely in the United States, or not, is really uncertain. I would simply say that this hope has been expressed before in a number of other contexts. Really, does this prediction materialize? And so I will conclude with where we started, namely with this infographic of the Nuffield Council on Bioethics, which identified in vitro-derived gametes as the next frontier, as the one that they will address first, before all the other topics we discussed earlier. So I'll leave you with this notion and conclude by saying, stay tuned, because at any moment now, nobody knows when, of course, we could all be treated to a science or cell paper from Japan or elsewhere, which reports the successful conclusion of in vitro gametogenesis in the human, be it the production of eggs or sperm, which would, as I suggested, likely transform reproduction as we know it, IVF included. So with that, I will stop here. If there is time for questions, I'd be happy to, of course, entertain them. But otherwise, I'd like to thank you for this opportunity to visit with you again. I believe I was with you about 2 years ago. And so I'm pleased to have this opportunity to be of service again. Thank you very much.